All right, everyone. It is 11 o'clock here on the East Coast, and I want to say good morning to everyone and officially welcome everyone to Virtual Visions 2020. My name is Jason Menzo, and I'm the Chief Operating Officer here at the Foundation Fighting Blindness. And I appreciate all of you for joining us from across the country and even from across the globe as we pivot visions to a virtual experience this year. I know hosting visions virtually isn't the same as being together in person, but I'm very excited for the program we've developed and sincerely hope that everyone enjoys the wealth of information that will be shared over all the sessions in the coming days. We have over 1,400 people that have registered for Visions, and this is the biggest number of attendees that we've ever had for a Visions. So in some ways, pivoting to virtual may be a benefit. And this session is streaming on Facebook Live and on YouTube, in addition to this closed interface that we're all using here in the session right now as well. Some of the sessions, like this one, will be live, and others will be pre-recorded and available on demand. For all scheduling details, you can go to our website, fightingblindness.org/visions2020. For the detailed schedule and all the information about the live and on-demand sessions. We are pleased to be closed captioning this session, and we also have an Asl Interpreter. And for attendees that need to view the Asl Interpreter, please use the 3 dots next to the interpreter's video to pin them to your screen. To activate the closed captioning, please select closed captioning at the bottom of the interface. And if you are using a screen reader, please be aware that the controls are at the bottom of the Zoom interface. The control bar may collapse when not in use. So if you'd prefer to prevent that, the controls from collapsing and from auto hiding, go to the settings within the Zoom interface and select accessibility, and then select always show the meeting controls. So now to get things started, I would like to welcome a very special guest speaker, our chairman of the board, Mr. David Brent.
Good morning. I am delighted to have all of you join us for Virtual Visions 2020 conference. I hope you're all well and you're safe and during these crazy times. And whether you're newly diagnosed with a retinal disease or have been part of the foundation for decades, we're family. And what's great about this conference is that after this 3-day whirlwind, I have no doubt that you will feel even more connected to our sight-saving mission, the quality of our team, and the dedication of the researchers we support. Over the next few days, you'll be inspired by all the great research you'll learn about. This is the result of almost 50 years of hard work by amazing researchers which we support around the world. And in case you don't know me, my name's David Brent, and my family's been part of the foundation for more than 2 decades. My son, Alan, was born with a condition called Leber's congenital amaurosis. And he is a college graduated on time, and he holds a job. And the poor guy, he was living in his own apartment, but with COVID, now he's been living with us for 3 months, and it's driving him a bit crazy, as you probably all know if you have kids at home. But you know, everything for him, in spite of his success, is just harder. And so, like most of you here today, we've had our own remarkable journey in dealing with retinal disease, and it is our journeys that connect us all. I also want you to know that the Foundation's journey, our path forward, is at a very critical juncture, but in a good way. Today, we have an incredible opportunity to accelerate therapy development, and we need you to be part and to continue to be part of that journey. Never before have we had so many emerging therapies in the development pipeline. There are promising treatments that are almost ready, and some are in human study, some are almost ready for human study. As most of you know, we had the first FDA-approved gene therapy for the eye, Luxturna, and there are about 40 clinical trials underway. There are 40 clinical trials underway. 40 clinical trials underway. For those of you who've been involved with the Foundation for a long time, this is amazing. When I first got involved, we knew 2 genes, and doctors said there's hope. Now we have 40 clinical trials underway. Furthermore, we have additional emerging treatments for Stargardt's and choroideremia and various forms of RP and achromatopsia and Usher's and LCA and AMD. And there are optogenetic and stem cell and other drug trials for treatments that are designed to work across genetic and disease profiles. So no matter what your disease is, these treatments will hopefully ultimately be able to restore vision. In the next 10 years, we have an opportunity to bring many more vision-saving treatments into and through the pipeline and across the finish line. Never before has so much vision-saving science been at our doorstep. What researchers need is an organization and a guiding partner to provide the money and the expertise, the industry resources, and the regulatory knowledge to advance these treatments to the point in which they're ready for clinical trials, ready for the biotechs to develop them commercially. And the Foundation Fighting Blindness is that partner. There's no organization on the planet with the commitment and required skill set. The fact is, researchers need our support to help scientific ideas and move them along, but they don't have the expertise or the money to clinically develop these therapies, to get them to people. And that's ultimately what we're doing. It's not a game of seeing how much science we can create. We want to help people. Companies are reluctant to invest in treatments if they haven't cleared some of these risky early developmental hurdles. That is where the foundation comes in. We're the bridge. We find the best research for emerging treatments. We fund the therapy development based on milestones, so we only pay for what they've accomplished. And we protect the researcher's intellectual property to ensure that there is an ability to raise the capital needed to do the clinical trials. We connect researchers with biotech and pharmaceuticals and for commercial development. The big challenge for the foundation is that each of these projects requires millions of dollars. So our revenue needs have grown exponentially. That's why we need your support now more than ever. We need you and your families to put the pedal to the metal. As we know so well, no one else is going to do it for us. Thank you. Enjoy the conference. Thanks for being part of the foundation. Stay well. Back to you, Jason.
Awesome. Thank you so much, David. It's been awesome getting to know you and getting to work with you, and your passion and leadership in our cause is second to none. So thank you. And thanks for kicking off Visions 2020 officially. What we're going to do is we're actually going to get things officially started with our very first session. This session is called Mission Possible: What's Next for Gene Therapy? This presentation will last about 45 minutes, and then we will have 15 minutes of the session reserved at the end for Q&A from the audience. I want to start with a few housekeeping notes. Before we get started, all of the participant lines are in listen-only mode. As I said a few minutes ago, today's session is being recorded. It is also live streaming on Facebook and on YouTube Live, and it is available in closed captioning. I mentioned a few minutes ago, this session also has an Asl Interpreter, and if you would like to view the Asl Interpreter, please just click the 3 dots on the screen next to their image and select pin, so that way they're always on the screen for you. To activate the closed captioning, please select closed captioning at the bottom of the interface. And if you are using a screen reader, please be aware that the controls are at the bottom of the Zoom interface. The control bar may collapse when not in use. And if you'd prefer to prevent the controls from auto-hiding, go to the settings within the Zoom platform, select accessibility, and then select always show meeting controls. I'll repeat this at the end, but just so you know, to ask questions during the session, you can use the Q&A feature on the Zoom control bar and type in your question. And all sessions during the conference will be recorded for you to access later. And finally, before we begin this session, I'd like to encourage you to be social with us. Share this experience using the hashtag Visions2020 on Facebook and Twitter. The speakers for this session are Drs. Shannon Boye and Dr. Ben Yerxa. And just a few words about both of them. Dr. Boye is an assistant professor of ophthalmology at the University of Florida. She is a world-renowned expert in the area of gene therapy for inherited retinal diseases. And she currently serves as principal investigator, co-principal investigator, or co-investigator on several federally and privately funded grants, and is actively involved in the University of Florida's teaching mission. Dr. Yerxa is the Chief Executive Officer at the Foundation Fighting Blindness, and as such is responsible for the overall management of the foundation. He also serves as the CEO of our recently launched RD Fund and has more than 25 years of experience in biotechnology, drug development, and translating promising research discoveries into clinical milestones and treatments. He also holds over 60 US patents. So with that, I'd like to turn the floor over to Ben and Shannon.
All right, thanks, Jason. And hello, everyone. It's great to be with all of you live today, and I'm glad to have such a big crowd joining us virtually. It's really, really great. We thought this topic of genetic therapies would be a good one to kick things off because of the amount of innovation and sheer variety of programs and approaches to each therapeutic challenge. Last time at Visions 2 years ago in San Diego, we were all talking about the Rna approval. And although there hasn't been another approval for an IRD or dry AMD since then, the R&D momentum we are seeing is astonishing, as you heard just now from David. And as I've said to some of you before, breakthroughs in medicine are usually not linear. They tend to be more exponential. And there's essentially a revolution happening right now in personalized medicine and genetic therapies in general. So I'm really delighted to have Dr. Shannon Boye here with us to walk us through these new technologies and select programs as examples. There are literally too many to go through one by one, which is a really good problem to have. So without further ado, Shannon, I'll hand it over to you.
Great, thank you, Ben. Can you hear me okay?
Yep.
All right, perfect. So thank you, everyone. Thank you to the FFB for having me here today. It's a genuine pleasure to be part of the FFB family. And while I'm upset that I don't get to see all of you in the hallways and at a reception, I'm actually getting text messages from some of my favorite IRD parents as I do this. So hello, Miriam. Shout out to Miriam. I'm going to put this down though so I can give my talk. I just want to say how instrumental the Foundation Fighting Blindness has been in my career. When you start out as a young scientist, it takes a lot to get your first grant, right? You're competing with very established scientists in the field with a lot of name recognition, and so you're just hopeful that someone's going to take a bet on you, and the foundation was who took that bet on me. And so I got my first grant from the FFB, and with that money I was able to generate the data that I needed to compete successfully for a much larger grant, and then that led to subsequent grants and a bona fide research career, and they've continued to fund my research since that the beginning. So I can't say enough how grateful I am to the foundation for helping me get on this path. So I want to go back, before I get into all of the developments that Ben mentioned, I want to go back to sort of high school and middle school level science and go over some important details so that you guys can follow along with sort of the rest of what I'm going to say. And I'm going to break it down into sort of a science way, and I'm also going to give you an analogy to hopefully follow along, and via either of those two methods, hopefully this message will be accessible. And I should say too, even though we can't meet each other out in the hallways, I'm always happy to answer questions via email outside of this forum. So one thing that's really important is that all of us have these little letters in our bodies, and those little letters are called DNA. And subunits of those letters, or that DNA, are called genes. And genes do something really important. They make something called RNA. So genes or DNA make RNA, and then RNA does something really important. It makes a protein. And a protein is really important. You can think of proteins like the building blocks of life. They act alone or in concert with a bunch of other proteins to perform essential functions. So imagine in your photoreceptors, in your retina, you have hundreds of proteins that all have to coordinate and act together in order to convert an incoming photon of light into an electrochemical signal that's then sent to your brain and processed there as vision. So again, DNA makes RNA, and RNA makes protein, and protein performs an essential function. So if you have a problem at any one of those steps, you can envision, no pun intended, how vision might break down because there's a problem and ultimately that protein can't do its job. So let's talk about that in more of like a silly analogy format. So imagine you're out and about. Let's pretend coronavirus isn't a thing. You're at work, your kids are at school, your brother's at baseball practice, but your mom knows that you're going to be the first one that gets home at your house that afternoon. and you've got a really cute lab at home that needs to go to the bathroom, needs to be let outside, right? So she sends you a text message that tells you to please take the dog out. You get that message and you realize, I've got to take the dog out. So you perform that function, you go take the dog out. So there's a bunch of letters that combine in a certain way on your mom's cell phone screen. She hit send and you see that message. You can think of that as the RNA. And then you are the protein performing the function when you take the dog out, right? So what happens if you have a problem with the letters? So what if your mom is trying to type you that message and her phone, there's something wrong and she can only get out 6 letters of the text. So all you see on your phone is the word please. You walk in the house, you see please. You're like, what does my mom want from me? You don't take the dog out. So in the end, the function isn't performed. On a more subtle level, there can be problems with the letters such that maybe your mom's phone doesn't produce the letter D. Whenever she pushes the D button, an L comes out. So you walk in the house and you see the message, please take the log out, instead of please take the dog out. So this is an example where one change in one letter can make a very different message. So not only do you think your mom is insane, but you don't take the dog out and then you have a mess to clean up. So I hope I'm conveying that letters need to be in a very important sequence in order for a coherent message to be generated, and then a message tells a protein to perform a really important function. And if there's a breakdown at any stage in that cascade of events, visual impairment can occur. But the good thing is that we have developed strategies in the research community to combat problems at each step of that cascade. And a couple years ago, like Ben said, there really wasn't much out there to talk about. In fact, I've given some of the talks at these early vision meetings where I was able to get through what was going on within just a few minutes. And as Ben said, there's no way that I'm going to be able to discuss all of the science that's going on today or all of the clinical trials even. And as he mentioned, that's a great problem to have. So I'm going to go through 3 different strategies that are being used to address inherited retinal disease today, and just give a sprinkling of certain indications that are being addressed via each of those methods. We're not going to touch today too much on delivery methods, but I will just say that all of the technologies that I'm going to be describing to you, for the most part, are packaged inside a viral vector called AAV, which we can deliver to the eye via different strategies. But again, if you have follow-up questions about that, please don't hesitate to reach out to me. So the first type of technology we're going to be discussing is sort of the easiest conceptually. It's called gene supplementation or gene replacement. And everybody's very familiar with Luxturna, of course, because it's FDA approved, but that was a gene supplementation or a gene replacement technology. And the basis of this is that you want to make sure that the right protein is expressed in the patient's retina. So you deliver the right letters that make the right message and then make the right protein. That's a pretty simple concept, right? That's Luxturna. And that same premise is being applied to address many other inherited retinal diseases. And so I'm just going to go through a few examples right now. First and foremost, and near and dear to my heart personally, is another form of LCA that's caused by mutations in Gucci2D or GUCY2D. This causes LCA1, and this is actually a more common form of LCA than the LCA2 form for which Luxturna now exists. And over the course of the last 15 years, we have established that gene replacement therapy works in animal models, in a number of animal models actually, and that we could restore retinal function and visually guided behavior to animal models of this disease. We were very fortunate in 2014 to align with Genzyme, which then became Sanofi, to push this program into the clinic. And this gets to what David Brent was saying, which is that there's a lot of expertise involved in sort of crossing what the NIH calls the valley of death, between where you have a ton of data, proof of concept in animal models, but then actually making it to the clinic. And so Genzyme Sanofi very much helped us with that. And we're excited to report that that phase 1/2 trial has begun. It's early days, so of course there's no publications, but this is an example of another, perhaps, the next Luxturna being right around the corner, excuse me. The next really good example, and also in the clinic, is a form of retinitis pigmentosa caused by mutations in RPGR. So this is called X-linked retinitis pigmentosa. And there's been a lot of great proof of concept work done by collaborators of mine at the University of Pennsylvania, Art Sidisian, Bill Beltran, et cetera. And I have to say, if I forget to name someone's name, just know that it's because of a time constraint, but there have been many, many people that have been involved in these efforts that I don't intend to leave out. But they've done some very seminal work to characterize a naturally occurring dog model of this disease. And having those large animal models is really crucial to understanding the disease and also being able to test gene therapies. And so they were able to test a gene therapy and show that it worked very well in the dog models to restore retinal function, preserve retinal structure, and restore vision to those animal models. And because of their work, and also because of Rob McLaren's work in a mouse model of the disease, there have been multiple companies that have taken this forward into clinical trials. This includes AGTC, which is using an AAV2-based variant to address the disease, and Biogen, which is using an AAV8-based vector. So they're using slightly different flavors of a gene therapy, but the goal being the same. And there have been some early indications that this is going very well via press releases from AGTC, as well as posters that were presented at this year's American Society for Gene and Cell Therapy meeting from the Biogen group. And they reported exceptional visual improvement and even some outer segment regrowth in one of the patients that received the high dose. So this is looking very promising, and it'll be interesting to follow. Another disease that is being addressed in the clinic is choroideremia. And again, there are multiple people who have been involved in the preclinical effort, including Ian McDonald, Rob McLaren, Byron Lamb, Jean Bennett. There's too many to name. But choroideremia is a disease that impacts both the photoreceptors and the retinal pigment epithelium at the back of the eye. And based on preclinical data, this also was moved into clinical trials by Nightstar, now Biogen. And they have published their results and shown improved visual acuity in some of the treated patients. So that's also been an exciting outcome. Another disease, X-linked retinoschisis, or juvenile XLRS. This is a condition that affects only males. It's an interesting condition in that it's characterized by what we call schisis cavities in the retina, where the photoreceptors literally become detached from the underlying neurons. And when that happens, of course, blindness can occur. And this has shown a ton of preclinical evidence of efficacy. And because of that, two groups, both AGTC and the National Eye Institute, both pursued a clinical trial for this disease. Both of them used an intravitreal approach. As I mentioned earlier, there are many ways to deliver these drugs to the eye, but intravitreal is especially attractive because it can be done in an outpatient setting. It doesn't require a full-blown surgery. And so those two entities pursued an intravitreal-based treatment for that disease. And unfortunately, there's some issues with intravitreal injection that you don't deal with when you're doing a subretinal injection. And so the immune system is more of an issue, put simply, when you deliver vectors to the front of the eye. And we learned a lot from those trials in that we need to pay attention to the immune system. So while we didn't see a super robust outcome necessarily from those first two trials, we learned a lot, and it's really going to inform the next generation of trials aimed at addressing that disease. Next, we have achromatopsia, multiple forms, of course. We have the CNG-A3 as well as the CNG-B3 forms of achromatopsia. This is a cone-mediated disease. Patients suffer from photophobia. They don't see colors properly because our cones are what we use to see color. But there was a lot of preclinical evidence suggesting that this would be effective in patients that was demonstrated by friends of mine, Andros Komarami, Stylianos Mikolakis, Ron Offrey, and there were a variety of animal models, dogs, mice, and sheep. So this was a great effort preclinically. And because of the promising results that were shown, AGTC and Mira GTX, as well as the University of Tübingen and University of Munich, have all pursued clinical trials to address these forms of achromatopsia. So the CNG-B3 form specifically is being addressed by AGTC and Mira GTX. We don't have any published results to date, however. The CNG-A3 form is being treated or has been treated by the University of Tübingen and Munich, and they have published their results and did show modest improvements in visual acuity, contrast sensitivity, and chromatic discrimination. So this is all very exciting, and I should mention also the CNG-A3 form is also being pursued by Mira GTX, but no publications yet. So you'll hear me say no publications yet a lot of times during this talk, but I think there's a lot that's about to come out, so that's really exciting. Best's disease is a little bit different in the sense that it's caused by problems at the level of the RPE, so not the photoreceptors. But what happens in this disease is that the interactions between the RPE and the photoreceptors, which are adjacent cell layers, and they sort of interconnect like so. But because of a mutation in a gene called bestrophin, that interaction is impaired. And so you get these big separations or detachments in the retina. And as soon as the photoreceptors come detached from the RPE, vision loss ensues. But there's been a lot of great work done by colleagues at the University of Pennsylvania, Karina Guzewicz, Gus Aguirre and their team, who have a very good animal model, a dog model of Best's disease, and they've established proof of concept for gene replacement in this model. And this has now been picked up by Iveric Bio, formerly Optotech, and is hopefully being moved into clinical trials soon. So that's another exciting development. Let's talk for a minute about something that isn't necessarily only related to the eye, but Bardet-Biedl syndrome, or BBS. This is a group of diseases that affects multiple body systems, and it's because it's related to defects in the cilia of the cells. And you have tons of cells in your body that have cilia. So if you have a problem with your cilia, you can understand why there would be multiple organ involvement. So patients with BBS typically have polydactyly, obesity, but they also suffer from retinitis pigmentosa. And so there have been efforts at the University of Iowa, for instance, by Arlene Drack, looking at gene therapy in mouse models of BBS1 as well as BBS10, which have shown promise. Similarly, efforts have been made with BBS4. And then excitingly, Martha Nuringer at OHSU has a naturally occurring monkey model of BBS7, which is going to be a very important tool in terms of understanding the disease and then also clarifying whether gene therapy will work in that condition. Last but not least, there's another type of gene replacement it's a little bit more complicated. What happens when you have a mutation in a large gene? Something that we haven't really discussed at length is that the AAV virus that we're using to deliver genes, like what was used in Luxturna, it's only a certain size. It can only accommodate about 5,000 of those letters that we talked about. But certain genes are too large to fit inside AAV. And for that reason, years back, people thought, well, let's just use a larger virus to accommodate that large gene. And two examples are myosin 7A Usher syndrome 1B and ABCA4 Stargardt's disease. So both myosin 7A and ABCA4 are large genes. And so initial efforts involved putting those large genes into a larger virus called lentivirus and delivering those to the eye. Unfortunately, lentivirus is not that good at infecting photoreceptors, but all was not lost. We learned a lot from those trials, but they're kind of in a holding pattern right now. But in the background, we've been developing what we call dual AAV vector mediated approaches to treat these two diseases. And put simply, what we do is we take this large gene and we split it in half. We put the front half in one AAV and the back half in another AAV, and then we co-infect those two AAVs into the same cell. And then via a region of overlapping sequence, they can recombine to form the full-length gene, then go on to make the RNA they're supposed to make, and then go on to make the protein that they're supposed to make. So there's some really strong preclinical data coming out of both my lab as well as Alberto Arriccio's lab in terms of myosin 7A. And then in terms of ABCA4 Stargardt's, both Bill Houseworth, Alberto Arriccio, as well as Rob McLaren have been working on this. And both of those are slated, hopefully, to go into clinical trials in the near future. And I should mention, too, that another way perhaps to deal with large gene diseases is to chop out some of the unnecessary sequence. And sometimes that works, sometimes it doesn't. But if you can make a large gene small enough to fit inside a standard AAV and it still works, great. And Iveric Bio is actually pursuing this strategy for ABCA4 Stargardt's disease, CEP290, LCA10, as well as a form of Usher syndrome called USH2A. So that is just a sprinkling of what's going on in the gene supplementation space. So again, that's dealing with the letters, okay, the letters at the DNA level. But what happens when we get to the RNA level? There are actually approaches where we're trying to fix things at the RNA level too. And we refer to this whole space as RNA therapeutics. So one form of RNA therapeutics is called antisense oligonucleotides. That sounds super scary and difficult to understand, but it's actually pretty easy. You can think of antisense oligonucleotides as the autocorrect on your mom's phone before she sends you that message, so that you take the dog out. You're not going to get a bad message. So if your mom's text message said, "Please take the log out," you can think of AON like an autocorrect. It's going to bind to that L and it's going to change it to a D so that it fixes the message. Then you get the correct message, "Please take the dog out," and then you go and you do it. So the function is performed. Antisense oligonucleotides are most often delivered by themselves, so not inside a virus. We say that that's delivering them naked, but because of this, they do require repeat injections. But there's a very exciting trial going on now to address a form of LCA, a very common form caused by mutations in CEP290. And this is based on preclinical work out of Rob Collins's group. And this is now in phase 1/2 clinical trials. It's being run by ProQR using antisense oligos to address a very common form of this disease. And again, it's too early, there's no publications, but the preclinical data suggests that this should be quite effective. Another antisense oligonucleotide program underway preclinically is from Jen Lentz's lab at Louisiana State University, and she's using AONs, or antisense oligos, to address USH1C, which is a form of Usher syndrome caused by mutations in a hormonin. So that's antisense oligonucleotides, but there's actually another older form of RNA therapeutics that has shown a lot of promises and is being taken into the clinic, and this is called RNA interference. So in this strategy, a small molecule called a small interfering RNA literally will bind to the bad message and eliminate it altogether. So instead of autocorrecting a certain letter in the message, it would be like Siri realizing that the text message, "Please take the log out," was so stupid that it just had to be erased altogether, and you never got the message at all. So the end result is there's no bad message and no bad protein can get made. But let's hope that you still know that you need to take the dog out. So this is especially useful for dominantly inherited diseases. And in a dominant disease, what happens is that you have a mutation in your DNA and a bad protein gets made. And it can be misshapen such that it doesn't interact properly with the other proteins in your photoreceptors. And maybe it causes a buildup of things in the cell that ultimately make the cell sick. It has some toxic gain of function. So in that scenario, it wouldn't be sufficient to do gene replacement, because in gene replacement, you're delivering a healthy copy of the gene, and that's going on to make a healthy protein, and that's all well and good, but you still have this bad protein over here that's causing a problem in the cell, and you need to get rid of that. So that's where RNA interference comes in. You can actually deliver what's called an siRNA to bind to that bad message and make that bad protein go away. This has been tried a lot in the retinitis pigmentosa space, specifically for rhodopsin-associated autosomal dominant RP. And there's been a lot of great work done by my colleague Alfred Lewin at the University of Florida to show that expression of mutant rhodopsin could be knocked down by targeting that bad message or that bad RNA. And in some cases, that's enough. You can just get rid of the bad RNA and rely on that other copy of healthy message to make the normal protein. But sometimes you have to knock out the bad message and deliver a good message. So it sort of depends on the scenario. But based on all of that exciting preclinical work, Iveric Bio has paired with Al Lewin and his colleagues to push this forward into phase 1/2 trials. So RNA therapeutics is another really exciting area. Finally, the third really exciting area, and perhaps the newest, is called gene editing. So this really gets to the root of the problem. This goes all the way back to the letters over here that your mom was going to arrange in a certain sequence. Because remember, the letters in our DNA have to be in the right sequence in order to make a coherent message. And we now have a way to rearrange those letters to ensure that the right message gets across. So this is based primarily on what's called CRISPR-Cas9 editing technology. This is actually something that exists in bacteria, if you can believe it. It's a natural defense system that bacteria have against viruses because they want to remember, hey, that's a virus invading me. I don't want you here. We don't have time to get into the science behind that, but I think it's pretty cool that we're using something for bacteria to have this, to revolutionize medicine the way that we are. But you can think of CRISPR-Cas9 like genetic scissors. And what we do in this system is that we use something called a guide RNA to literally drag a special enzyme to a region of the DNA that we're interested in. And usually that region would be the region that contains the mutation, or the L instead of the D, for instance. So we guide that enzyme to the DNA, and then that enzyme, which is called Cas9, literally cuts the DNA. And there are many, many different variations on this theme. We can simply cut the DNA sort of early on in the sequence and then prevent it from ever making a message. Or we can go in and cut out a very specific area of the DNA and replace it with the right letters so that a coherent message gets made. So it's a very hot area right now. It's really new technology, but it has really opened the door to addressing a variety of inherited retinal diseases that perhaps we weren't addressing before. So very exciting. There's a form of LCA, a very common form caused by mutations in CEP290 that is actively being treated in the clinic right now with the CRISPR-Cas9 based technology. So this is an AAV5 delivering a CRISPR-Cas9 reagent to address a very common form of this disease. And it's officially called Edit 101, and this is being sponsored by Editas Medicine. So again, the first patient was dosed within just a few months ago, I believe. So we're anxiously awaiting the results of those trials. A similar approach is being taken by Editas Medicine for USH2A. They have exciting preclinical data showing that this would be possible. We don't have this in clinic yet, but hopefully that will be the case. And there's also a new form of gene editing. It's sort of the new kid on the block, and it's called base editing. And the difference with this strategy, without getting too complicated, is that instead of your Cas9 enzyme coming in and cutting both strands of your DNA, it cuts one strand, and then another really cool enzyme comes in and switches one letter to another. It's all very, very refined and neat. But this has opened up the door to even more genetic editing success. And that's a strategy that's also being applied to USH2A. And I should mention USH2A is one of these very large genes that we can't fit inside a standard AAV vector. So a gene editing approach for that form of Usher syndrome is worth pursuing. A similar base editing / gene editing strategy is being employed for RP1 associated retinitis pigmentosa. This is preclinical, not yet in the clinic. Another form of gene editing being pursued is something called HITI, or homology independent targeted integration, which is a lot of words, but all you need to know is that we can cut the DNA and put really large pieces of the normal gene back into the patient's cells. And this is an approach being taken for ABCA4 Stargardt's disease by Ivana Trapani in Italy. The same approach is being taken by Alberto Arricchio for autosomal dominant retinitis pigmentosa caused by rhodopsin mutations. And then finally, in my lab, we're pursuing a gene editing based treatment for cone rod dystrophy 6, or CORD6, associated with mutations in Gucci2D. And the goal there is to knock out the bad copy of Gucci2D and replace it with a healthy copy. So again, in the interest of time, there's no way I can go through all of the things that are going on in the field right now. That was literally the tip of the iceberg. And so again, I'm happy to answer questions outside of this, but there's just no way to cover it all. But what's important to understand is that, like Ben said, there's an absolute exponential increase in the therapies that are being developed. And importantly, the technologies and the techniques that I just described to you, so gene supplementation, RNA therapeutics, and gene editing, in no way are restricted to the disease indications that I mentioned. They can be much more widely applied to a number of genes and a number of indications, and that's happening at a variety of levels in the field right now. So this is an incredibly exciting time to be a retinal gene therapist. I feel like I have ADD, like I want to cure everything. I want to use every approach that's out there. But but I hope that the summary provides those of you in the audience that maybe don't have RP65LCA2 that one day there's going to be a Luxturna for your inherited retinal disease too, and just assure you that we're all working very, very hard to do that. So with that, I really want to say thank you to Ben and to Jason and David and to all the members of the FFB for everything that they've done for me. I feel like we've become a family of sorts, and I really get a lot of personal satisfaction out of the relationship that I've developed with the foundation and all of the families that I've met through your organization. You guys are the best. So thank you for your time, and I'm very happy to take questions.
Thanks, Shannon. That was really, really awesome. Catch your breath. That's a lot of stuff to go through, but hopefully that was a really good overview for everyone who's tuned in. A few things I just want to point out from the foundation perspective is that if you look at essentially every major program, either in the clinic or approaching it, the Foundation Fighting Blindness has been essentially an integral part in making it happen. And not every single program, but pretty much the majority of the programs, we've been a part of it. And fighting blindness, you know, we really cast a wide net in our funding strategy. And we can do that because our therapeutic focus is so narrow. And the strategy is really paying off. You know, as you've heard, dozens of programs in the clinic now, dozens more approaching the clinic in the next 1 to 2 years. And essentially the foundation has moved the entire field forward with its concentrated funding strategy. And you should all be really proud of that progress. Long-term supporters and people who are getting involved. You know, it's really because of you that we're able to make this happen. So enough bragging about the foundation. Now we really want to move on to some questions. And we see some questions kind of coming forward. And I have a few that I can use to kick things off. So let's see. So maybe Shannon, I could ask you one question to kick it off, and then we'll kind of see what's in the queue from the chat. So since, you know, this personalized medicine is really taking off, not just in the eye, but pretty much in all areas of medicine, we're starting to see kind of new issues or different kinds of IRDs getting more attention in terms of, like, you know, Friedreich's ataxia and Batten's and others. And maybe you could just speak to that a little bit, since I know where you are at University of Florida. You also, with gene therapy, Powell Gene Therapy Center there, you see kind of what's going on with some of those other diseases and some opportunities in the eye.
Yeah, I think this is a really exciting and important direction that the field is embracing, and I'm excited that the foundation is excited about this as well. But bottom line is that there are a variety of diseases that affect multiple systems in the body, right? And the most logical approach to addressing those types of diseases is to develop a systemic gene therapy. So something you deliver intravenously to help things like the heart or the kidney or the liver or whatever might be sick. But the problem with that is that the vectors that you deliver systemically never really reach the eye at high enough efficiency to have a therapeutic effect there. And unfortunately, in some of these diseases, like Friedreich's ataxia or Batten's disease, you do ultimately have a vision component. And so we really have to start thinking about these patients that are going to receive these systemic gene therapies. What's going to happen in the later stages? How are we going to address the ocular phenotype that hasn't been necessarily corrected by that systemic gene therapy? Or simply just a later stage patient that hasn't even received the systemic gene therapy, but still wants to benefit from improved vision. So we're focusing pretty heavily now on that aspect. So taking a vector that can be delivered intravitreally to a Friedreich's ataxia patient, for instance. This is a disease that affects the mitochondria. And one of the most metabolically active cells in our body are the retinal ganglion cells. So they have a ton of mitochondria. And so they're especially impacted by that disease. And lucky for us, the retinal ganglion cells are the first cell that the AAV will hit if we do an intravitreal injection. So we think there's a high likelihood for our ability to correct the visual phenotype. And maybe we can give that vector in conjunction with the systemic gene therapy or alone in later stage patients. So my lab is personally pursuing that effort. There are also labs pursuing efforts on Batten's disease, which is a group of what we call neuronal ceroid lipofuscinosis diseases. That's a mouthful. But these are diseases that affect the entire central nervous system. And again, even though we can deliver an AAV to the brain, it's not going to get to the retina at a high enough efficiency to fix the visual phenotype. So there's a lot of exciting work coming out of Missouri from Rebecca Whiting, who's looking at a CLN2 based dog model of Batten's disease. There's also a naturally occurring CLN7 monkey model that was identified by Martha Neuringer at OHSU. So I think we're really on the cusp of diving into that field where we're going to start looking at fixing the visual phenotype of these larger diseases. I think it's a really exciting area.
Wow, great answer. It's really, really comprehensive. Thank you. Jason Menzo, I think you're pulling some questions together. Do you want to kick us off?
Thanks, Ben, and thank you, Shannon. Just for the audience, I want to remind everyone, so we'll have about 15 minutes, 15, 20 minutes or so. There's, a ton of questions that have come in, which is awesome. So we'll sort our way through the questions that have come in, but just to remind everyone that to ask a question, to please use the Q&A feature on the Zoom control bar and type your question in the interface there. And we're going to try to get to as many questions as we possibly can. But what I will say is that a lot of the questions that have come in are very specific to an individual's circumstances. And what we'll do in those cases, or really for any question that we can't or don't have time to get to in the session, we'll follow up with you via email one-on-one. And for any questions that you have that you didn't chat in, you can always contact us at info@fightingblindness.org. So just a couple of reminders there. Ben, actually, I'm going to address this question to you. There's a number of questions that have been chatted in about how to enroll in a clinical trial or how the foundation can help facilitate matching patients that are interested in clinical trials and the industry sponsors that are exploring them. So maybe you could speak a little bit to that.
Yeah, good question. I think first thing to do and think about is making sure you're in the registry. So the My Retina Tracker registry is really an important way for people who are interested in being in a clinical trial to be contacted and notified of those opportunities. And if you've been in the registry in the past and you think it's been a little bit static, well, things are really getting ready to change. We've changed our provider, made a lot of upgrades, and there's a lot going on clinically. So if you're interested in clinical trials, definitely get into the registry so you can be contacted when those opportunities arise. Second of all, go to our website and look to see what programs are in the clinic, find out which companies are involved. And usually you can go to the company websites and there's a patient portal where you can get more information and see if you qualify. And then if you ever have any issues with this or are just confused about it, just reach out directly to the foundation. As you've heard, go right to our info inbox and we'll help you out.
Awesome. Thank you so much, Ben. I'm super impressed. There's a number of questions being chatted in from folks from Brazil and from Spain and from the Far East. So it's kind of cool to see the global exposure we're having here today. Shannon, I'm going to try to combine a couple questions into one. There's quite a few questions about the difference between gene augmentation type of approach and gene editing. There's a couple questions about is Luxturna the same as CRISPR? A lot of these terms are in the media and folks hear these words, but maybe you could speak a little bit to the differences between these approaches specifically.
Sure, no problem. I get this question from families that have actually had their kids treated with Luxturna, so it's still confusing even at that level. What is gene supplementation versus what is gene editing and CRISPR? So as I mentioned, gene supplementation, which Luxturna was, is just delivering letters, DNA or a gene, into a patient's cell. Those letters then make RNA, and then that RNA makes the protein that needs to be there. So you've literally delivered or supplemented that gene back into the patient's cell for it to go and do its job. Whereas gene editing is you are delivering elements inside a virus that can go in and literally cut the DNA. You can think of them like genetic scissors. So you're not putting in a gene, you're going in and you're cutting the patient's own genes.
Very good. Thank you for that. That was very simple.
Sure.
It's straightforward. It's fantastic. This next one, I'm gonna address back to you, Ben. So a lot of folks are curious about the timeline and maybe at a 30,000 foot view, what happens from the point in time where something is identified in a lab, gene therapy or really otherwise, and from that moment to the point in time where it's FDA approved and in the patient's hands. So obviously it's a lengthy and really detailed process, but maybe you can share at a 30,000 foot view some of the key milestones for folks.
Yeah, it's a good question, and one we get often. And essentially every program's unique. But I can say in general terms that if there's a therapy that's being developed where there's already good proof of concept in a good animal model, and that program is fully funded, it should be able to get to clinical start in about 24 months. Sometimes it can take 3 years because of the manufacturing part can be pretty complex for gene and genetic therapies. So I'd say 2 to 3 years from that really solid animal result. And then you're on a pathway where it could take probably another 5 to 7 years to get to final approval. But it just depends. Sometimes these programs can go fairly quickly. But when you're doing gene therapies, you have to follow patients for a long time because these are one and done. They're not simple, and you have to proceed very cautiously in the beginning to make sure that you're not doing any harm. So it's, again, about 2 to 3 years to get into the clinic, and then another 5 to 7 to get it finally approved.
Awesome. All right. Hopefully that helps folks. And again, anytime that there's a specific question beyond what we're covering here today, you can always reach out to us at info@fightingblindness.org. There are certainly more questions that have been chatted in than we'll have the opportunity to get to in this session, but we will follow up with everyone on email afterwards, just so if we don't get to your question here today, don't be disappointed. We will follow up with you. Shannon, there are a number of questions that are specific to the diagnosis itself. And there's a number of folks asking about, and this is maybe a little bit more of a clinical question than a treatment question, but maybe at a high level, you can describe some of the differences between dry age-related macular degeneration, wet AMD, and Stargardt's. And one person in particular and I think that this is kind of a frustrating thing, because I've heard this from a number of folks, that they've gotten a diagnosis of Stargardt's, and then they get a diagnosis of RP, and then they get, over the course of their, 10 years, they get different clinical diagnoses from different eye care professionals. And so it's kind of a combo question I'm going to put in front of you from a clinical diagnosis standpoint. You could maybe speak to that.
So full disclosure, I'm a mouse doctor. I'm not a people doctor, but I will answer this to the best of my ability. But yeah, I think that this has been one of the frustrating things in the field, but it's something that's changing very quickly because there's been so much more attention paid to the therapies being developed for inherited retinal disease. But 10 years ago, you would bring or you would go to the ophthalmologist, and they might diagnose you with age-related macular degeneration simply because they didn't know that you've had Stargardt's disease for the last 20 years. And the bottom line is that there's a lot of commonalities between a lot of these diseases, and it's sort of like a rose by any other name. It can be called one thing or it can be called another thing, but the bottom line is it's a very common phenotype. But in terms of what's the difference between the two, Stargardt's disease is considered a juvenile form of macular degeneration. So it's going to involve the same area of the retina, in other words, dysfunction and degeneration of the central cone cells in the macula, but it's going to happen much earlier in life than age-related macular degeneration, which, as its name implies, is associated with age. So it's a disease that affects the elderly. And then in terms of differences between wet and dry AMD, wet AMD is characterized by a breakdown in the blood-retinal barrier, so neovascularization, so new vessels growing into the eye, which can unfortunately leak, and then when that blood leaks, it can lead to immediate vision loss in certain areas of the eye, whereas dry AMD doesn't tend to involve new vessel growth, rather just a buildup of materials sort of at the back of the eye. So there are subtle differences between some of these diseases, for sure.
Very good, thank you. Maybe I could ask Shannon one other sort of treatment-related question. Number of questions about, again, this is not specific to a particular gene as much as maybe the clinical stage, be it early stage versus late stage of a particular disease progression, and whether or not different potential treatment strategies are more geared towards early stage disease versus late stage disease, where gene therapy may have a role as compared to other strategies. Perhaps you could speak to that.
I think it's less about age of intervention when you're thinking about what technique should be applied. I think the technique that's applied is dictated by the underlying genetic mutation, not so much on the age of the patient. So certain diseases or mutations are going to be more amenable to gene supplementation, whereas other diseases may be more amenable to gene editing. I don't think it's fair to say that gene editing would work better than gene supplementation in a young patient, for instance. So it's really specific to the genetic mutation that you're trying to deal with.
Ben, I'm gonna turn a question over to you about the genetic testing in particular. You mentioned my Retina Tracker a few minutes ago. There's been a number of questions about folks that are asking about, I've been genetically tested. They hadn't been able to identify my gene. Should I be genetically tested again? Different tests, maybe around just the differences between different tests. Now you mentioned it, maybe you can go a little deeper into that.
Sure, I can try. Of course, this is a better question for Brian Mansfield, our Chief Scientific Officer. But in general, we obviously are a fan of the panel that we are running as part of the My Retina Tracker program. It's a free genetic test to anyone diagnosed with an IRD. So if you haven't done our panel, you might consider that if you haven't had a genetic test in the last couple of years. There are some specific criteria around that Brian could explain in a different session. Sometimes you don't get a full answer. So I forget what the exact percentage is, but not every single gene has been identified yet. So some people may not get an answer. But if you've had a test that's probably more than a couple years old, the panels have been getting better and better over time. So you may be eligible for a new modern panel that's free.
Very good, thank you. And again, if anyone would like to go deeper on any questions that are asked, you can always send us an email at info@fightingblindness.org. I'm now scanning somewhere in the neighborhood. Yes.
Can I throw a question over to Shannon?
Please, yeah. I was gonna say, I've got to read like 65 questions.
So Shannon, I've seen some questions coming in about later stage disease, where there's not a lot of photoreceptors left, and these people may not be good candidates for straight on gene therapy, but there is optogenetics. And I wonder if you just had a few words to say about how optogenetics works and what that might mean.
Yeah, absolutely. So in a normal healthy retina, the only cells that can actually respond to light are the photoreceptors. They have all of the proteins inside of them that are required to respond to a photon of light and convert it into an electrochemical signal. But unfortunately, in a lot of inherited retinal diseases, you lose those photoreceptors. And things like gene supplementation aren't going to work if your photoreceptors aren't there, because there's no cell to deliver that healthy gene to. But some really brilliant people have come up with something called optogenetics. And the basis for this is that you're essentially going to make the remaining cells in the retina that weren't by themselves actually light sensitive. You can actually make them light sensitive. And you do that by delivering essentially a channel protein that can react to light. And so it's forcing remaining cells that are still in retinas after the photoreceptors have gone away to be light sensitive. And so there are a variety of efforts going on in the field, including by some FFB funded companies to what they say reanimate the retina. So even if they don't have photoreceptors, to somehow allow for those remaining cells to be light sensitive and send a signal to the brain that can be processed as vision.
Great. Yeah, I think it holds a lot of promise. It's definitely it's a moonshot kind of approach, but it can potentially work in a gene independent way. So we're excited about that and funding it further. maybe one other question about sort of later stage disease and cell therapies. And I know I'm kind of putting you on the spot. You're not necessarily an expert in regenerative medicine, but just from your perspective as a scientist, any thoughts on progress on regenerative medicine?
Again, you said it correctly. I'm definitely not a cell therapist, more of a gene therapist, but and so my bias is towards gene therapy. I think that that field has moved a little bit faster, but cell therapy certainly is showing promise. There's really exciting science that people are doing to subretinally inject progenitor cells into the subretinal space that will ultimately make connections with the retinal cells that are there and then be able to restore vision. There's other people that are trying to turn Mueller glio, which is a type of glial cell in the retina, into a photoreceptor.
Right.
So there's lots of stuff going on in the space, and I think I'm kind of waiting on bated breath to see what happens with it, actually.
Yeah, the field's a little bit earlier than gene therapy,
Yeah,
for sure.
yeah,
Yeah,
for sure.
Yeah.
But exciting nonetheless.
Yeah, fair enough. Jason, do you have another one, or do you want me to go?
You know, there has been a number of questions about a variety of the BBS genes. And Shannon, maybe you could speak to that. I'm scrolling through, and again, for those who are participating online, there's so many questions, and so it's fantastic to see the engagement. We will follow up with any question that isn't answered here live on the session offline, and so don't be discouraged if we don't get to your question. But I do see a number of questions specific to BBS1, BBS2, and maybe you could speak to your knowledge on that, Shannon.
Sure. So again, just for the audience, Bardet-Biedl syndrome, or BBS, it's a group of ciliopathies that affects multiple body systems. But there is the possibility to address the visual impairment in BBS. This isn't my area specifically, but I do know that there have been some proof of concept studies done in mouse models of BBS1 as well as BBS10 that are showing promise. And then a few years back, there was also a paper from Sam Wu's lab looking at proof of concept in a BBS4 mouse model. And then again, as I mentioned, there's an amazing resource at OHSU, at KCI Institute. They have a monkey colony, and they purposely look for naturally occurring mutations in that colony, and they've identified one that has a BBS7 mutation, so a form of BBS. And so while I haven't seen any therapeutic results come out of that model, I think it's very, very early days, but that's going to be a really powerful tool in terms of understanding that form of BBS as well as testing therapies. And I don't know, Ben, if you know of any other BBS efforts out there.
No, I think those are the major ones. Yep,
Okay.
Thank you.
Mm-hmm.
Awesome. And then the final question that we'll do here live, there's also a number of folks that have asked a variety of questions around Usher syndrome and all the various types. And maybe we can end on just a brief overview, Shannon, from the work that's happening in the area of Usher syndrome.
Okay. And Ben, chime in on me, because there's a lot of a lot of Usher's genes. But what's near and dear to my heart, obviously, is myosin 7A Usher syndrome 1B. As I mentioned, there was a lentiviral-based program to try and address this form of Usher syndrome, which is sort of in a holding pattern right now. But we have been evaluating a dual AAV vector-based approach to address this disease, and the same is true of Alberto Arriccio's lab in Italy. So I do think that within the next couple of years, we're going to see a dual AAV vector-based approach for USH1B in the clinic in phase 1/2 clinical trials. As I mentioned, USH1C, this is a program being pursued by Jen Lentz's lab, and she's sort of taking a multifactorial approach. She's evaluating both antisense oligonucleotides as well as gene replacement for that program. Then there's USH2A, which is, of course, one of the most prevalent inherited retinal diseases. The problem being is this is a very large gene, so we have to be very smart about how we approach developing a treatment for USH2A. And there are multiple strategies being pursued there. So Editas Medicine is looking at a gene editing based approach. Iveric Bio is looking at sort of a mini gene approach, making it smaller and delivering it with a standard AAV vector. There are new base editing approaches, which is sort of the newfangled CRISPR-Cas9 system. So there's a lot of interest in USH2A right now. Ben, do you have any more USH genes?
Well, I think what I'll add there is that as a foundation, we're funding a large natural history study called RUSH2A. And even though it's focused on the larger, more prevalent USH2A, we're learning a lot about how to do these natural history studies for ushers in general. And I think that'll help pave the way for some future natural history studies. And what we found is that when we fund natural history studies, it spurs industry involvement because it helps them understand endpoints and timelines and things like that that allow them to de-risk their clinical development programs.
Oh, one other I forgot to mention, a form of USH3 caused by mutations in CLEAR, and there's a lot of good preclinical work out there in that program as well.
Yeah, great, thank you.
Mm-hmm.
Perfect. Actually, I know a second ago I said we take that as our last question. There's two other topics that have been sort of coming in fast and furious, so we'll maybe address those two. Shannon, I'll come to you in a second for a similar, maybe 30,000 foot view of the Stargardt's landscape. But first, maybe I'll ask you, Ben, to speak about what's on the horizon for my retina tracker. A number of questions about, because we do have an international audience, about any plans to take it globally, or just sort of what does the future look like in terms of enhancing the My Retina Tracker and genetic testing. So maybe we'll start there and then we'll wrap up with Shannon speaking to start.
Sure. Yeah, I mean, I think we'll have more on this later in the program. But basically, we've got a new provider that allows us to have more interactions with people who are in the registry. So, the future of the registry from our perspective is that it's not just a touch point to identify people or to connect people with trial opportunities, but we may also be able to send out questionnaires and things like that so that we can start to generate real world data to the people who are signed up. And that's really important information for people who are developing therapies. So, stay tuned on that. I think that's really important. Otherwise, we're gathering some really important sort of epidemiological information by collecting all these data together in a very private and secure way. We look forward to understanding that better as well. Other than that, take advantage of the free testing that's available right now. We're trying to find ways to make that continue for as long as possible. Right now it's free and it's the best test out there. So take advantage of the opportunity to get a free test and to get into the registry. And as we progress with our ability in the registry, I think you'll see a lot more contact, a lot more opportunities to contribute to the field as an affected individual.
Awesome. And Shannon, do you want to put a bow on this session with just speaking from a genetic therapy standpoint of advancements to Stargardt's from again, a 30,000 foot view?
Sure. Yeah, and I know there's a lot of interest in this because it is a fairly common IRD. And I think there's a lot going on in this space right now, utilizing multiple techniques that I described earlier. So from the gene replacement or gene supplementation standpoint, the challenge that we face with Stargardt's is that it's a large gene. It can't fit inside a single AAV vector. But as I mentioned, we've developed an approach where we can split the large gene in half, deliver one half via one capsid, the back half via a second capsid, co-infect those capsids into the same cell, and then full-length ABCA4 forms. It goes on to make the RNA and make the protein. And so these dual AAV vector-based approaches have been characterized nicely by Bill Houseworth's lab, as well as Alberto Riccio's lab. And they will likely be pursued. And I believe Iveric Bio is pursuing exactly that, as well as AGTC. So from the gene supplementation standpoint, there's a lot of promise for Stargardt's. There's a lot of preclinical work using gene editing as well. I mentioned something earlier, kind of a mouthful, but it's called HITI, or Homology Independent Targeted Integration, where folks are essentially taking that CRISPR-Cas9, coming in and cutting the DNA, and then inserting this huge piece of ABCA4, a huge healthy piece of ABCA4. And they're doing that sort of with the front half of the gene as well as the back half of the gene. And in so doing, they're addressing mutations that exist all the way across this very large gene. And that's in the preclinical stages. It's being done by Ivana Trapani in Italy. And so I will watch that closely for any effects that it might have. So I think, we're doing both gene supplementation as well as gene editing to find ways to address Stargardt's in the clinic. And I think that there'll be a lot of new data coming out soon.
Awesome.
And Jason, maybe I could add just some of the other programs that are going after the vitamin A cycle. some programs in phase 2 and phase 3. So I think we have to stay tuned on that. Iveric has a complement program that's also in a fairly large phase 2 trial in Stargardt. And Alkeus has a program in phase 2 with a deuterated vitamin A. And then there are other companies looking at ways to moderate the visual cycle and reduce the levels of vitamin A circulating as another potential treatment.
Awesome. All right. Thank you so much, Ben. Thank you so much, Shannon. And thank you to everyone who has been on the line and for all the many questions. And I'll reiterate again that we did not get to every question that was asked in the session. That's okay. We will follow up with folks individually offline in the coming days and weeks ahead. And at any time, if you have any questions about subject matter in this session, or really anything related to the inherited retinal disease landscape or age-related macular degeneration, don't hesitate to reach out to us at info@fightingblindness.org. I'll also, just drop my cell phone, sorry about that. I'll also use this as an opportunity to plug the quarterly Insights Forum calls that we have that are an opportunity for Ben and others in leadership here at the foundation to provide regular updates once a quarter on things that are happening in the IRD landscape. So this is not the only time and opportunity to ask questions or to engage in this type of subject matter. So be on the lookout for announcements in future Insights forums. And the other thing I would mention is that over the course of the next several days, there's a ton of additional content. So a lot of the questions that were asked in this session may actually be answered in some of the other sessions that are planned. And there's a ton of content that is on demand that is available right now. So as soon as we end this session, you could go right to fightingblindness.org/visions2020 to stream some of the pre-recorded content. And then obviously all the live content and moderated panels like this one that are planned over the next couple of days. So with that said, I encourage everyone to join us for our next session that is going to be this afternoon at 4:30. That session is around advancing cell therapies and cell-based approach to treating IRDs. And again, for all details related to Visions 2020, be sure to go to www.fightingblindness.org/visions2020. Thank you all, and we'll see you in the next couple sessions. Bye-bye.